Okay, this is a very controversial topic for a video. Before I proceed, I want you to understand a few things. The first one is to understand my intention. I am not saying that cannabis cures cancer. The word cure is commonly used and abused in common conversation. I'm not even recommending that you use cannabis for cancer. Everyone is different, everyone's disease is different, everyone's treatment plan is different. There are so many different types of cancer, and it is up to you, your doctor, and your oncologist to decide what's best for you. So don't extrapolate what I'm saying, but the preliminary preclinical evidence for cannabis and cancer is pretty interesting. So I'm going to show you some of the recent advances as it pertains to cancer and cannabis right now. So just for some background, our endocannabinoid system has become a fancy new target for the treatment of cancer. Why? Because cannabinoid receptors play a role in tumorigenesis, the creation and growth of tumors. They also play roles in the inhibition of tumors and in metastasis, which is cancer spread. But cannabinoid receptors are often upregulated in things like tumors. And the upregulation and expressiveness relates to things like tumor invasiveness. Some authors have even detailed how loss of CB1 receptors can lead to accelerated growth. So while given that a lot of the things I'm going to show you are pretty incredible, none of them should be really unexpected. First, let's tackle skin cancer. The study I want to look at is cannabidiol and minor phytocannabinoids that was published in the Journal of Pharmaceuticals in 2023. This study looked at melanoma, a common skin cancer. This type of cancer originates from melanocytes, so these are the cells that create melanin. Now, melanoma can be difficult and also resistant to treatment. And this is why researchers are looking at cannabis as a method to slow or eliminate its spread. So, that Polish study found this. They found that cannabinoids do have an effect on melanoma cell growth. A375 melanoma cells, which are a specific line of human skin cancer, were pretty sensitive to cannabinoid treatment. Cannabinoids reduced cell viability, and furthermore, it was dose dependent. So this means you add more cannabinoids, you get more of an effect. So it appears that cannabinoids interfere with the internal machinery of a cancer cell. They trigger what is called apoptosis. So this is programmed cell death, which is of course needed in cancer treatment. Now these results are preclinical, meaning that they just occurred in the lab, and in particular cells, not directly in human beings. We would need clinical trials to prove the efficacy and safety. However, this is early stage research and more is going to be needed. So this study implied that cannabinoids, particularly CBN, can inhibit melanoma in a few ways. All right, now that we've established that, let's look at a cancer that is strongly female-specific, breast cancer. Breast cancer is unique not just because there is an expression of cannabinoid receptors, but other receptors as well. Some tumors overexpress the protein HER-B2, also known as ERB-B2, or HER, substitute as you will. HER2 breast cancers are usually super aggressive, and they're also very likely to spread or metastasize. Let's take a cannabinoid like THC, and even synthetic versions of it like JWH133. There is an impression that activating CB1 receptors that could respond to something like THC could influence cancer cell signaling through pathways like AKT. Now, the Journal of Molecular Cancer published a study on breast cancer progression when we influence something like AKT. They looked to see whether or not cannabinoids could influence breast cancer tumor growth and metastasis in ERB2 positive breast cancer. This study is unique in that it combines animal models, cell culture experiments, and analyses of human tumor samples. So how did they do this? Specifically, the researchers looked at MMTV-new transgenic mice. This is a specific type of mouse that will naturally develop those HER2-positive breast cancer tumors. And these mice were treated with that JWH133 synthetic cannabinoid. And they monitored tumor size, tumor progression, and tumor number over time. What they found was that mice that were treated with THC and JWH133 had significantly reduced tumor size and number. All of this when we compare them to the controls. The tumor progression was far slower, and some of them even showed signs of tumor death. What it shows is that in HER2-positive breast cancer, cannabinoids can play a role in ensuring that tumors spread less rapidly. Now, mechanistically, this was of course because of that AKT signaling pathway. You see, AKT normally promotes things like cell survival, cell growth, and cell proliferation. By inhibiting it with cannabinoids, they're less likely to grow. Think of AKT as kind of like a switch in a cancer cell. Now, a little bit of something for the future. Analyses show that 91% of ERBB2 positive breast cancer expresses CB2, which means that most patients with this subtype could likely respond to some sort of cannabinoid therapy. That's pretty incredible. So, let's summarize. Cannabinoids such as THC or JWH133 can inhibit tumors. This is done through the AKT signaling pathway, and in the future, this could be a therapeutic target. So Schaffarel and co. in that Journal of Molecular Cancer showed that this has potential. Now we can move on to a common form of brain cancer, and that is glioblastoma multiforme, which from now I'll call GBM. Now, GBM is an aggressive, treatment-resistant form of cancer, and it has a very poor prognosis. Among the cannabinoids, CBG, cannabigerol, has garnered a lot of attention as a potential therapeutic treatment for GBM. Now, a 2021 study from the Journal of Cells employed a combination of in vivo and in vitro approaches to see if there was anti-cancer effects of CBG on GBM. They treated glioblastoma lines with CBG and looked at apoptosis, cell viability, and proliferation. A glioblastoma mouse model was created by injecting these cells into the craniums of mice. Western blotting was done, and that one's for protein, by the way, as well as immunohistochemistry. And they looked to examine the key proteins that are essential for the survival of the cancer. Now, what did they find? First thing, they did find apoptosis, and they found that CBG initiates this. Secondly, they found cell cycle arrest. They found that CBG arrests the cell cycle at G0 to G1, which means the cancer cells have a tough time dividing. And here's the big one. They also found that there was no toxicity, which means that normal tissue survived. Cannabinoids had a selectivity for cancer cells. So let me give a quick explanation for non-scientists, less wordy. Cannabinoids made these cancer cells self-destruct. They also prevented their cell clock from allowing them to divide. In mice, tumors grew slower and cancer cells showed cell death. And the cannabinoids were not toxic to any other cell but the cancer cell. So this is compelling evidence that shows CBG could have pronounced effects on cancer cells, particularly glioblastoma. However, like all of these, more studies are needed. Rats and mice is promising, but humans will be the next big step. And lastly, let's take a look at prostate cancer. A 2023 study in the Journal of PLOS1 explored the anti-proliferative effects of CBD, cannabidiol. This is looking at the PC3 line of prostate cancer cells. PC3 was cultured and treated with varying amounts of CBD, and the cancer cell viability was examined. This was done with flow cytometry and Western blotting. Simply put, what they found was CBD treatment led to a significant decrease in these cancer cells' viability, and this had a maximum reduction of 37.2%. Also, CBD treatment led to DNA fragmentation in those cancer cells. But one of the major takeaways is that gene expression even changed. There was upregulation of a certain apoptotic gene called BAX. So CBD actually made the cancer cells express themselves differently and express themselves to potentially die. So this is great, this is promising, but I'm not going to stop there. We need to talk about a lingering question. Sure, these cannabinoids could help with somebody who is dealing with cancer, potentially. But many people who are dealing with cancer aren't just using cannabinoids, they're using other pharmaceutical drugs. Now, could they interact, the cannabinoids and the pharmaceuticals? Someone with prostate cancer would use a chemotherapy like etoposide or something. So could cannabis work well in concert with that medication? Well, a 2023 study in the Journal of Cannabis and Cannabinoid Research investigated the combined effect of CBD and etoposide. They cell cultured prostate cancer cells and treated it with CBD or etoposide or both. They looked at cell viability, protein expression via Western blotting, and flow cytometry. They also used quantitative PCR to look at gene expression. Now, what did they find? They found that in combination, CBD and etoposide had enhanced cancer cell cytotoxicity. So CBD and etoposide had a greater effects than each of these alone. They didn't interfere with each other. They led to increased DNA fragmentation, so increased apoptosis and death of the cancer cells. And gene expression also changed. There's a higher expression of pro-apoptotic genes. So this shows that CBD can potentially work synergistically with existing chemotherapies for prostate cancer. So something as innocuous as CBD has the potential to affect cancer cells. It could end up being a valuable component, but of course, more studies are necessary. But to reiterate what I said earlier, all cancers are different because you're different. This isn't just a matter of the location of the cancer or the effects of the cancer. On a genetic level, they all differ. Cancer research with cannabis is still in its infancy, but like an infant, it is quickly growing, and cannabis has the potential to help make people's lives better as it relates to cancer. There are many more studies like the ones I've mentioned available. I've just scratched the surface. But in doing so, I hope I have shed some light on the potential therapeutic benefits of cannabis as it relates to cancer and cancer treatment. Dave & A provides individualized, customized cannabis care and consulting for people like you or me. People who could be dealing with some of the issues I've mentioned here today. Saying take one marijuana daily is not good enough, and it's never been. So let us